Hello, hope you're well. So this is a brief summary of DNA replication, not to replace the lecture, but just to act as an aide memoir for your revision purposes. So DNA replication, as you know, involves double-stranded DNA, which is tightly wound together, and the process by which we unwind that double-stranded DNA, and then we make two new double-stranded DNA molecules. The diagram that we can see on the slide shows that double-stranded DNA, which you can see in a blue color here. The double-stranded DNA at the start of the process is called the parental DNA, which I'll just highlight for you here. This is the parental DNA. Parental DNA is made of two strands. You can see them here, the 5' to 3' strand. I'll just highlight them there, the 5' to the 3' strand. You can follow that all the way around, and the 3' to the 5' strand. Each of these individual strands acts as a template for a new strand to be made. See that highlighted on here, the two template DNAs. So how do we take that double-stranded DNA? How do we make new nucleic acid material? We do that using an enzyme, and that enzyme is called DNA polymerase 3, which you can see here, this purple kind of circular structure. And DNA polymerase 3 elongates nucleic acids. So it needs something to start it, then it takes 3 DNTPs and starts making new chains of nucleic acid. Stack. How does it start? We'll see that in a minute. It needs something to start on. It can't begin on its own. So DNA polymerase 3 is the active enzyme here that elongates the chains. And there's a couple of slides in the lecture which talked about that process and showed you the molecular detail that goes on there. So how do we go from a double-stranded DNA molecule, which is tightly bound with each other, to something that allows these two template strands to be read? We do that with the help of two enzymes. The first of these is DNA Helicase, highlighted here, and shown in this little kind of pucey color. So DNA Helicase, in the lecture we talked about it being like a zip. So if you imagine DNA is very, very tightly bound like the zip of a jacket, the actual zipper itself goes down and pulls that apart. Well, DNA Helicase does the same. It unwinds the double-stranded DNA, revealing the two template strands and allowing the rest of the process to take place. The second enzyme is the DNA Primase, which you can see here and highlighted in this little green color. And the DNA Primase lays down the first few bases, the first few nucleotides of the new strand. This allows the DNA polymerase, it gives you something to attach to, so then it can carry out its elongation function. That's what it does. Just the kind of way you'd prime a wall before you put the top coat of paint on it. It gives you the preparationary layer. And you may remember from the lecture that these primers are actually RNA, not DNA, and of course we need to get rid of those later on. And they're shown in the diagram as this little green material. You can see several of them further up. So the helicase unwinds, the primase lays the first steps, then DNA polymerase 3 combine to start the elongation process. Now the two different template strands from the original DNA, the two different ones, where are we? The two different ones have slightly different things happen on them, and that's because of the direction of the template strand. So on the 3' to 5' strand, you can see perhaps from the diagram that the new material shown in a purple is all one continuous line. This strand is called the leading strand. It's called that because DNA polymerase 3 works in the 5' to 3' direction. That's the direction it works, the gear, the shift, the way that it works. And because it's reading a 3' to 5' strand, kind of making the opposite, it can do that in one continuous motion. Whereas on the other strand, we've still got the DNA polymerase working in the same direction, but here the template is 5' to 3', the direction with which the polymerase can work or make. So this is a problem, and you may be able to see just from where this little arrow is that the polymerase is actually kind of pointing backwards. So what happens? Effectively, we make the primer the first step, then polymerase will work backwards as this arrow indicates here. Meanwhile, we can see that we've got the same process happening further upstream. And again, a new one is just starting here. So effectively, we're making a series of short fragments, all working kind of in the opposite direction to the overall direction of replication. And these need to be, these little fragments, which are called Okazaki fragments, you see the word highlighted here, will need to be joined up. Each of those fragments has their own little RNA primer, which you can see here. So after these fragments have been made, or as they're being made, DNA polymerase 1 will remove the RNA primers, and DNA ligase will stick those pieces back together to make one continual strand. Now this strand is called the lagging strand. You can see that up here because of its speed. It lags behind the other strand because of this kind of stagnated jump approach it needs to take. We can also see on here some single-stranded binding proteins highlighted here. These are these little purple dots you can see. These help stabilize the lagging strand specifically and stop the single-stranded nucleic acid forming any interactions or bonds with itself, because stabilize it while this process takes place. The final element to perhaps just show you is the this. So if I just draw a nice triangle here to show you. So if I do that, this is the overall direction of DNA replication. And this element here is called the replication fork, kind of the active area. That's because this looks very much like a fork, a pitchfork, a devil's fork, rather than an eating fork. So that's called the replication fork. So that's the overall process, and fundamentally this is in prokaryotes, so simple organisms, bacteria. We also talked about eukaryotes, so you need to understand the differences and the similarities between those two processes. And we looked about some specific areas such as at the end of eukaryotic DNA and telomeres. So I hope you find this useful. Thanks.